Hello, APBIO. In this video, I'm going to talk about the matter processing system. And I'll start just by talking a little bit about matter. Remember that matter is just all the things on the periodic table. Everything in the whole universe, whether it's living or non-living, is made of matter. Living organisms are a very precise organization of matter. It takes a tremendous amount of energy. I'll talk about the energy processing system in the next video. It takes a tremendous amount of energy to organize matter, but life is just a precise arrangement of matter. Now, the matter is arranged by a system of interconnected membranes that are all in, well, kind of direct contact. I'll talk about not really direct contact in a minute. Some of them are in direct contact. Others are sort of are connected by a shuttle. In any case, the membranes are inside cells and they're all interconnected. So because they're inside cells, we call them endo, meaning insides, the endomembrane system. So the endomembrane system is the matter processing system. And you can think about the matter that is processed through the endomembrane system. It follows the endomembrane. I think about it as a path. Now, just like when you might walk on a path, I don't know, to someplace, you might make pit stops and have a picnic in one place and set up your tent in another. But you might not. Maybe sometimes you don't eat a picnic, or maybe sometimes you set up your picnic in a different spot. So it's important to say in the very beginning that you might not make all the stops. So just like you might not make all the spots when you're walking along the path, matter doesn't make all the stops. Also, matter can move back and forth through the endomembrane path. And so it's just important to say that the path is not necessarily unidirectional. Parts of it are unidirectional, and I'll talk about that when that's the case. And also parts of it matter must pass through certain parts, but not all parts are unidirectional, meaning some parts go back and forth. Some stops along the path might be stopped at more than one time. So I'll make note of that when that's the case. But again, we're talking about a system of interconnected membranes. Let's take a second, go to your notes, and label the different stops on the path.
Okay, so here we are in your notes, and I'm here where it says all cells must process matter. So we're talking about the matter processing system. And you see here it's called the endomembrane system. And let's actually write that down, what I was just talking about, that not all matter has to stop at all stops. So let's say that we'll call them substances because we're talking about molecules and other types of chemical substances. They may not stop at all locations along the path, and they also may move in both directions sometimes. So they can move back and forth along the path. There is a notable exception, and we'll talk about it in a second. Now let's go ahead and label all the different stops along the endomembrane system. And I'll talk to you a little bit about how the notes are arranged. The first stop on the endomembrane system is this right here. This is the nuclear envelope. So we can go ahead and label it on our diagram, and we can give it a little 1 right here. Here is the second stop in the endomembrane path. This right here is the rough endoplasmic reticulum. We'll go ahead and label that. And let's do that and so on and so forth all the way through the path. This right here is called the smooth endoplasmic reticulum. And then the fourth stop on the endomembrane system isn't labeled. We're going to draw it in. We'll put a little vesicle in right here. In between this smooth endoplasmic reticulum, which is right here, this area right here. This is called a transport vesicle, and we'll put that in right there. I'm going to write a slash. I'm going to come back to that in just a second. The fifth stop along the way is this right here. This is the Golgi apparatus. We'll go ahead and put that in. Now, the Golgi apparatus is not in direct contact with the plasma membrane, which is the next stop. So we're going to, these right here, these little budding, little tiny compartments that are coming off. We can even put in a few more if we want to. This is number 6 right here. And those are also called transport vesicles. That's why we have 2 numbers right there. And then number 7 is right here. This is just the plasma membrane. So we'll go ahead and put that number in too. And I'm gonna talk about lysosomes and vacuoles in a different video. Now, just like we did in the processing system, information processing system, we're going to talk first about the biochemistry. So again, we're going to try to connect it to our first unit. And then we're going to talk about the structure of the subcellular components and then the function of the subcellular components. So the anatomy and the physiology of the endomembrane system. So we'll start first with the nuclear envelope. The nuclear envelope is just made out of a biological membrane. So it's a lipid bilayer, just like all membranes are lipid bilayers, but has this really complex protein pores. So they're made out of protein, and there are these holes that are going to make this particular membrane very selective. Again, it's think about it. If you think about it, it's protecting the genome, so it's important that it's very selective. Now, we call it the nuclear envelope and not the nuclear membrane to bring attention to the observation that it's actually a double membrane. So it's 2 layers of lipid bilayer, and there's a space in between the 2 membranes that's called the inter, for in between membrane space. And that intermembrane space is continuous with the whole ER, which is the next stop, which is number 2. So this space in between here, which you can't even really see in this diagram, is continuous with the space here in this second stop on the endomembrane system. Now, the purpose of the nuclear envelope is it of course protects the genome. But if we're talking about it in reference to the endomembrane system, it's going to connect the nucleus with the matter processing system. So now the nucleus has this sort of direct secret tunnel to the matter processing system. Now, the next step on the endomembrane system is the rough endoplasmic reticulum. And the rough endoplasmic reticulum is going to be the lipid bilayer. Again, it's just a bunch of membranes. And it has proteins and RNA. The reason that I say that it's the lipid bilayer with embedded ribosomes. That's why it's called the rough endoplasmic reticulum. And the endoplasmic reticulum is just this network of membranes. So it's like this secret tunnels, and that has these like different membranes that are all folded on one another. And in the case of this part of the endoplasmic reticulum, because you'll notice in a second we're going to talk about more endoplasmic reticulum, this has attached ribosomes. If you look up here in this diagram up at the upper right of your notes, this is an electron micrograph of the endoplasmic reticulum, and you can see this folded membrane in here. This is all folded membrane, and then these little dots, those are all ribosomes. Now, the purpose of the function of the rough endoplasmic reticulum is that it really is the ribosomes that are performing the function. Remember that they, from the information processing system, these make polypeptides. Now, if polypeptides are manufactured on the endomembrane system, they're specific. The polypeptides are either going to be embedded in membranes, so they can be used in the cell and embedded in membranes, or they're going to be exported out of the cell. So thinking about what the endomembrane system does, it's moving matter, and there's no way to get proteins or substances out of a cell unless it's part of the endomembrane system. The endomembrane system is connected out here to the plasma membrane, and you'll see in a minute, if a cell needs to get rid of something, it has to be the substance has to be somehow along this path of the, or the has to be somehow along the endomembrane path. Now the third stop on the endomembrane system is here, and I've kind of colored it funny, so I'm going to use a different color so you can see it. It's here. This is the smooth endoplasmic reticulum, and the only reason I know that it's the smooth and not the rough is because there's no dots on it in this cartoon drawing that I have right here. And the difference between the smooth endoplasmic, it's still just the lipid bilayer. Again, it's a bunch of membranes. But it's going to have these specific proteins that are specific for the endoplasmic reticulum. So we'll say that it's a lipid bilayer plus proteins, but it has no ribosomes. So let's say that right here, no ribosomes. Now, both the rough and the smooth endoplasmic reticulum, I'm going to give you a little bit more terminology to be able to talk about how the membrane is folded. We use the term cisterna to refer to these, they're called tubules, and then there's some sacs, these sort of irregularly folded areas that are enclosed by the folded membrane. We also use the term lumen, and we'll use that word quite a bit when we talk about cellular anatomy, to refer to the space inside the endoplasmic reticulum. So you can think of that as the internal space. And that internal space is very important. The biochemistry of that space is very important. Now, the smooth endoplasmic reticulum has a really variable function. It really depends on the type of cell that it's in. Sometimes the purpose of the smooth endoplasmic reticulum is that it synthesizes more lipids. Notably, it makes phospholipids, part of the process of making phospholipids, to make more membranes. It also is going to metabolize carbohydrates, all right, carbs, and it's responsible for detoxifying toxins. So cells that are responsible for cleaning things up are going to have a lot of smooth endoplasmic reticulum. For example, your liver is responsible for detoxifying if, for example, alcohol, and liver cells have a lot of smooth endoplasmic reticulum. We'll talk a little bit about tolerance in class after, when you're back in class.
You can see here an electron micrograph of the endoplasmic reticulum. You can see how highly folded it is. And this shows you the difference between the rough and RER, and the SER. This dark right here is showing you the ribosomes. If you look down here, you can see it sort of in the cartoon. You can see how it's all just one continuous. So it's all basically the same stuff. The ribosomes are attached to the endoplasmic reticulum that is adjacent to the nuclear envelope. And if that makes sense when you think about it, the job of the ribosomes is to make the polypeptides. The RNA, which we'll learn about protein synthesis further along in the semester, but the RNA are coming directly from the nucleus. And so those ribosomes are attaching directly outside the nucleus. Why would they travel farther? You can also see that it's sort of this system of secret tunnels. I think of these I think this is called a Sharpay. This is what I think of when I think of the endoplasmic reticulum. It's all this folded membrane, which is important because it provides not only space on the inside, the lumen, that's what this term refers to, but also all this surface area for all reactions to happen.
In order for substances to get from the smooth endoplasmic reticulum over to the Golgi, little tiny membrane compartments will bud, it's called budding, bud off the smooth endoplasmic reticulum, little taxi cabs. And so they come off of the endoplasmic reticulum and then they sort of fuse into the Golgi apparatus. So number 5 is the Golgi apparatus. Now, we call that little tiny little membrane that buds off a vesicle, and it's specifically called a transport vesicle. So let's write down a little bit about transport vesicles. First of all, it's just a lipid bilayer plus proteins. And if you see so far, these are all just membranes, so they're all going to be the same thing. There's going to be the biochemistry is just lipid bilayer proteins because they're just biological membranes. But the transport vesicle is just, you can think of it as a little mobile, little taxi cab, a little mobile membrane bound compartment. And the transport vesicles are going to deliver little packages, substances to the next stop in the path. And often that is step 5, which is the Golgi apparatus. Now, the Golgi apparatus is what I colored in here in yellow up in here. The Golgi apparatus is shown here. Now, remember I said, here's an electron micrograph of the Golgi. I said that some components of the endomembrane system are unidirectional. This is the case with the Golgi apparatus. Materials are always going to move only in one direction, and you can see with this arrow right here, I'm showing you the direction that they're going to move in. You can see here, these are little, up in this area in the micrograph, these are little transport vesicles. Here's a larger transport vesicle. And the Golgi apparatus is this series of flattened sacs. So it's almost like, I think of them as like pita bread. And let's describe it here. First of all, it's a lipid bilayer plus proteins because it's just a bunch of membranes. Those are biological membranes. And the one step up from that, looking at the anatomy, it's a series of flattened sacs. Now, different than the other parts of the endomembrane system, the sacs are actually not in direct contact with each other, but they sort of fuse together and then materials will move through. Now, the area of the Golgi apparatus that receives substances that are matter that's moving along the endomembrane path is called the cis face. And then the materials will move through and then they're released from the trans face. So you have the cis to the trans faces. Now, the purpose of the Golgi apparatus is going to modify proteins. And it modifies other big macromolecules too. It puts together carbohydrates and proteins together to make glycoproteins. It puts together lipids and carbohydrates together to make glycolipids. It's going to synthesize different macromolecules. But it's also going to package and ship and sort. So it's going to pack and sort matter. So you can think of the Golgi apparatus as sort of like a FedEx. It's like a pack and ship. And proteins need to know where to go next and how to get to the next place. And are they going to be exported or are they going to be put into membranes? And how are they going to get where they need to go? And who do they get to be packaged with? And that's what the Golgi does. It's going to pack and ship along with make a lot of the macromolecules. Now, this is step 5. If we go back just one step to go think about where we have to go next, the Golgi is not attached to the next, not physically attached to the next step on the endomembrane path. And so here we've got these transport vesicles again. So let's go back to those. These deliver and they also receive substances. So now the Golgi apparatus is going to dump some stuff into these little taxi cabs, and then they're going to move. So they're both going to deliver and receive. That's step number 6. So we're here, back up here. And those transport vesicles are going to move along motor proteins within the cell.
Vesicles are important because they're going to provide continuity along the endomembrane path. So we've got them working to move materials from the smooth ER to the Golgi. You can see these little dots right here. And then afterward, from the Golgi to the plasma membrane. So in both of those places, stop number 4 and stop number 6, the Golgi is connected to the smooth ER by these little transport vesicles. Not directly connected, but connected, dynamically connected, I think of it as. And then also the Golgi apparatus is dynamically connected to the plasma membrane. So here's a little bit more detailed view of looking at the receiving and the sending sides of the Golgi apparatus. Materials are going to be received from transport vesicles coming from the smooth ER, maybe directly from the rough ER. And then it's called maturing through. Substances will mature through the Golgi apparatus, and stuff in this sac will dump into this sac, or there's all kinds of theories about how materials actually move through. It's actually not particularly well understood. There's several theories. In any case, and then materials are going to bud off the trans face and then move to the next stop on the endomembrane path.
Let's go back up for a second and let's look at this electron micrograph for just one more sec, just to look at that cis and trans face. If I were to label this electron micrograph, here's the cis face up here, and then here's the trans face of the Golgi. And you can see those flattened sacs, or each one of these is a flattened sac, this long thing in here. And so materials are going to move in, come in the cis face and go out the trans face. It's just one of the exceptions to the materials moving back and forth across the throughout the endomembrane system. Now, if materials are going to be exported from the cell, then they have to make it all the way to the plasma membrane. And making it all the way to the plasma membrane is the very last stop. So we can go ahead and write down that the plasma membrane is just the lipid bilayer. Sorry, I don't like that one. It's too sharp. The lipid bilayer plus proteins. And now that we're talking about the plasma membrane, we can talk about, I just want to give you the terminology for the current theory that explains the current theory of membrane anatomy. That theory is called, or that model is called, the fluid mosaic model. Fluid mosaic model. And it's called the fluid mosaic model because of the biochemistry of the structure of the membrane. The fluid, we'll talk about it in chapter 7. The fluid portion of it is made up of phospholipids that actually are fats that are sort of this greasy, viscous, really viscous solution. And then the mosaic refers to all the different integral proteins. So these are the integral. Remember, we talked about the integral proteins, I don't know, the other day, I can't remember. Well, the purpose or the function, the physiology of the plasma membrane, we have a term that we use to refer to it. It only is going to let certain things into the cell, and at the same time, it's only going to let certain things out of the cell. We call that concept or that property the selectively permeable. So the membrane is selectively permeable. Now, really small molecules or small substances that are non-polar are going to be able to move through the fluid portion of the membrane. Think about it. It's non-polar, and they're pretty close together, but if things are small and non-polar, they can diffuse right through the phospholipids. If materials are not small and non-polar, if they're anything else, then they need to move through proteins. And the different proteins in the different membranes actually makes up the selective part, really, that the differences in membranes, the differences between membranes, so like all these different membranes as part of the endomembrane system, they all have different proteins in them. And that's what allows for selective permeability. Oh, this is going to let this thing in, and this is going to let something else in, and this other thing is going to let even something else in.
Okay, that's it for the endomembrane system, and I hope that was helpful.